Hello, my name is Bharti Parihar, and I am a grad student in the Almeida Lab. And the title of my research project is The Dynamics of Plantlet Development in Kalanchoe marnieriana. The genus of Kalanchoe consists of approximately 150 species and is found mainly throughout South and East Africa, Southeast Asia, and Madagascar. With the genus as part of one of the most diverse groups of plant families known as the stonecrop family, or Crassulaceae, diversity within the genus itself is no exception. Certain species have become popularly sought through consumer markets, where the likes of Kalanchoe blossfeldiana, which you can see in the top left here, are in high demand. This ornamental hybrid is desired for their small flowers and variety of colors, and their popularity is so prevalent that they can be found at most nurseries in areas with mild climate, such as here in California. Others are commonly found at local nurseries, such as Kalanchoe degramontiana, Kalanchoe marnieriana, Kalanchoe fezzianchoe, and Kalanchoe tubiflora. This project is divided into three aims. The first aim will focus on tissue level organization of axillary meristem establishment and plantlet development in Kalanchoe marnieriana. The two experiments associated with this aim is stereoscopic imaging, where that will be observing plantlet development throughout many days, and then scanning electron microscopy, or SEM imaging, which was performed at the UC Berkeley Lab Electron Microscopy Lab. The second aim will tackle the molecular underpinnings of the mechanisms underlying plantlet development in Kalanchoe marnieriana lateral meristems through a comparative transcriptomic approach. The three experiments associated with this aim is a differential gene expression analysis during the early stages of plantlet initiation, a comparison of genes that are expressed at the shoot apical meristem, or SAM, and non-meristematic leaf regions. And then finally, a total number of genes expressed analysis across species within Section 2, which is the section that Kalanchoe marnieriana belongs to, as well as some other species. The third aim will study genome size variation across Kalanchoe, with a particular focus on Section 2 species, and how this might correlate to reproductive modes. The two experiments for aim 3 is flow cytometry and Kamer counting. The stereoscopic imaging is complete of aim 1, and what was observed was there were significant developmental days, and these days have been defined as day 1, 5, 7, 9, 11, and 15. During these days, there was significant change phenotypically with these plantlets. This particular experiment informed me for my stereoscopic imaging, the scanning electron microscopy imaging, and those same significant days were then analyzed more closely with a scanning electron microscope to look at the morphology as plantlets developed through those days. The transcriptomics portion, or aim 2, the RNA extractions for this are complete, as well as the sequencing, and all that remains is the data analysis. With aim 3, the flow cytometry is complete. These are the associated genome sizes estimated through flow cytometry for each of the species, majority of them falling into Section 2, with some of them falling into Section 3, and a categorization still unknown for one particular species. The sequencing for Kamer is also complete using the ion torrent sequencing platform, and bioinformatics still remains for this particular experiment. I'd like to acknowledge, of course, my committee, Dr. Almeida, Dr. Baysdorfer, and Dr. Hazelhurst. CSR, of course, for supporting me throughout my project. The Berkeley Electron Microscope Lab for all of the SEM work. And, of course, the Almeida Lab members within the Almeida Lab for their general support, with a special focus to Dennis and Val, who have played a pertinent role in extracting for me the DNA and RNA. How the CSU East Bay Master's Program has shaped me. This program at CSU East Bay is really great if you want to pursue your PhD. It really lays down the groundwork for whether that is something that you want to pursue. And in my case, it is absolutely something I want to pursue within plant biology. There's a lot of foundational knowledge that you learn that you have to know in order to not just defend, but to move on to your PhD program in general. And of course, I had a wonderful mentor, Dr. Almeida, who was a key player in teaching me critical thinking. That is something that has to be taught. And you have a great mentor, you're likely to be taught great critical thinking as well. And that is the end of my presentation, and I hope that you enjoyed it.